Swayam Prabha. Digital India. Educated India.
So yesterday, I think around the time I was explaining about this K-cat, that is the rate constant for the rate limiting step. When we think of enzyme catalyzed reaction, these reactions have intermediate steps. Like for example, S doesn't become P, S combines with the enzyme to become ES, and that is an intermediate step. And when the ES becomes EP, that is another intermediate step. Then you have the EP dissociating as E plus P. So among these intermediate steps, whichever is the slowest becomes the rate limiting step, and the total rate of the reaction would depend on that particular step, rate of that rate limiting step. So the rate constant for that kind of a rate limiting step is what we call as K-cat. And the K-cat is also known as the turnover number because it basically indicates the number of substrate molecules converted into product per single molecule of enzyme when the enzyme is saturated with the substrate concentration. So now what we are going to do is we are going to consider how well K-cat and Km tells about the performance of the enzyme, which is technically called the catalytic efficiency. Yeah, so our goal is to have a parameter that accurately reflects the performance of the enzyme. So meaning how well the enzyme functions as a catalyst. So that depends on what is the rate acceleration over the uncatalyzed reaction. Okay, so that is not indicated by K-cat. K-cat is simply the turnover number. So the main point here is the turnover number for two enzymes can be same, meaning K-cat can be the same, but one enzyme may be catalyzing several orders more than the uncatalyzed reaction compared to the other one. So which means the first enzyme in this example is doing better than the second one, but that is not indicated by the K-cat. K-cat is simply the turnover number. And similarly, when you look at Km, many enzymes or most enzymes, their Km value depends on naturally is the given substrate low abundant or high abundant in the cellular environment. So the enzymes acting on substrates that are low abundant in the cell usually have lower Km. Otherwise, they are not going to be useful for converting that substrate into product. And compared to the enzymes that work on high abundant substrates where the Km is relatively higher. And that difference in Km does not reflect on actually the performance of the enzyme, because Km may be higher, but then the substrate is also abundant. So it doesn't matter that the Km is higher. So due to this, neither K-cat nor Km accurately reflects on the ability of the enzyme as a catalyst. Okay. So therefore, we use a ratio of these two called the specificity constant, which actually more accurately reflects the enzyme's performance. So if the catalyzed reaction, if the turnover number is high, then you're going to have a high K-cat. And at the same time, the same enzyme can work at a low substrate concentration, that is at a low substrate concentration itself. If it can reach half B max, then that would be Km. So then the ratio will be higher. So higher the ratio, better the performance of the enzyme. And this reaches a maximum that is limited by the rate at which molecules diffuse in the cellular environment. Like for example, the rate at which the substrate diffuses within the cytoplasm and become available for the enzyme, that becomes the limiting step. So that is something on which enzyme can't do anything. So environmental factors like the diffusion rate becomes the limiting. So if an enzyme's specificity constant is at the limit of the diffusion rate, then you say the enzyme has actually achieved perfection. So so here, as given by this equation, V0 depends on the concentrations of these two. It is actually a second order rate. This K cat by Km is a second order rate constant. So here are some of the enzymes and their substrates, and the corresponding K cat, Km, and the ratio are given. And if you look at it, the diffusion control limit is in this range, and some of these enzymes are already there. So most enzymes are actually limited only by the diffusion rate within the cellular environment, indicating many enzymes actually have achieved catalytic perfection. So this is not surprising given the very long time the life has been around and the evolution has perfected many of these enzymes. Okay, so after that we discussed about the the fact that most enzymes work on two substrates. So far we were looking at a single substrate to understand the steady state kinetics. So now we are going to look at not in detail about the kinetics of these bisubstrate reactions. We are just looking at what are the different reaction mechanism possibilities exist. Okay, one where the enzyme combines with the substrate in either order. Okay, S1 could bind first or S2 could bind for second, and then you have the ternary complex formation. Or instead of being this random, like S1 or S2 can bind first and the other one binding second, it may be ordered where one substrate binds first and then only the second substrate binds to form the ternary complex. This is called ordered reaction. So these are the two ways random order and ordered ternary complex formation happens. And some of the enzyme reactions involve no ternary complex, where the substrate 1 binds and gets converted, and then you have the first product released, then the second enzyme binds, and then second substrate binds, then that gets converted and then product release happens. So yesterday I used this ATP glucose itself is an example to explain this. For example, S1 you imagine is ATP, then enzyme ATP complex, then the terminal phosphate gets transferred to one of the active site amino acid on the enzyme. For example, serin's hydroxyl group can be phosphorylated. So now the phosphate is temporarily on the enzyme, and as a result the enzyme is now altered. It has an additional phosphate group, so you call it E prime, and the product one is the ADP. Okay, phosphate is transferred to the enzyme, so it became E prime. Then ATP has become ADP, and that's what is P1. Now that is released. Now to this enzyme phosphate you have the glucose binding. Then the phosphate group from the enzyme is transferred to this, so E prime now becomes E. And because S2 gets phosphorylated, it is no longer glucose, the substrate. Instead, it is glucose 6-phosphate, the product, and the product is released. So it can be like this with no ternary complex. And fortunately, the difference between whether it involves a ternary complex or not can be readily recognized using the kinetics that we saw so far. So this will be the way the double reciprocal plot or the Lineweaver-Burke plot looks for an enzyme that involves ternary complex formation. So here what actually has been plotted is each individual line you already understand, that is reciprocal of the substrate and velocity are plotted. The additional lines are because we have increased the substrate 2 concentration. So you take one particular concentration of substrate 2 and then vary substrate 1 and then you have one line. Then you increase the substrate 2 concentration, then you have the second one. Okay, and so on. So each of these lines correspond to one particular concentration of the substrate. In this particular direction, the substrate 2 concentration has been increased. Okay, so increasing the substrate concentration has reduced the slope here, meaning Km by Vmax, which is the slope. Km by Vmax equals the m in that equation that we saw 2 days ago. So indicating that presence of the second substrate affects the Km as well as to Vmax, because this also changes. Okay, and this non-parallel lines indicate ternary complex formation. And in a case where you have this situation where presence of substrate 2 will have no consequence on E binding with S1, because only E prime can bind S2. So S2 concentration is not going to affect this. And due to this, you get parallel lines for the situation where there are no ternary complex. Okay. The Km by Vmax does not change at all. So only the velocity itself, like this alone changes. Vmax individually changes because V0 changes. That is because this availability can drive this reaction this way. So therefore, it can speed up the ES breaking down. Okay. So due to that, Vmax decreases. Okay. So the main point here is, by looking at these lines, whether it is like this or like this, one will be able to tell whether ternary complex is involved or not. So here you are not really doing any molecular characterization of the protein. Simply by measuring the rate of the reaction, you are able to get an idea of what kind of a complex formation happens. So that's the goal of doing kinetics. So next we are going, so this is all we are going to learn about the enzyme kinetics, a basic introduction about how measuring rate of the enzyme catalyzed reaction can tell about some of the aspects of the enzyme itself. So next we are going to consider kinetics of enzyme inhibition. So inhibitors are molecules that inhibit the rate at which an enzyme converts a substrate into product. And many of the pharmaceutical drugs, the medicines that we take, they are actually enzyme inhibitors. Most of them that are currently in the market are all enzyme inhibitors. For example, the ubiquitous painkiller aspirin is an inhibitor of an enzyme called cyclooxygenase that prevents the formation of prostaglandin biosynthesis, hormones involved in the immune response in that particular context. So many drugs that we take are all inhibitors of enzymes. So therefore, kinetics of enzyme inhibition is an important branch of enzymology, and it's a good idea for us to learn at least an introductory level. So let us look at what kind of inhibitors are there and how they can be distinguished kinetically. The first one we are going to look at is competitive inhibitor. So the competitive inhibitors compete with the substrate to bind to the active site. So if an enzyme is bound with substrate, then it doesn't bind a competitive inhibitor, because both of S and I are competing to bind with E at the same place, that is the active site. Okay. So due to this, if you have a certain level of substrate concentration and if you increase the I, then ES formation gets decreased. Conversely, if you increase the substrate to saturating amount, that is what you will do to get to Vmax. Vmax is the maximum velocity that is attainable. At such a high concentration of substrate, the inhibitor gets competed out. So due to this, Vmax does not get affected by competitive inhibitor. So when you look at the graph, non-changing Vmax indicates, oh, this is a competitive inhibitor. On the other hand, the inhibitor competing with a substrate to bind enzyme, in a sense, reduces the affinity between the enzyme and the substrate, which means Km is going to increase. You need a lot more substrate otherwise more substrate to bind with the enzyme when the velocity is half Vmax in the presence of inhibitor than in the absence of inhibitor. So as a result, Km is going to increase, and because Km is going to increase, slope is going to change because slope is Km by Vmax. So as you increase the inhibitor, the slope is going to increase, indicating that the Km is increasing. So this slope change due to Km increase, but the ordinate intercept not changing, indicating Vmax not changing, is a hallmark of competitive inhibition. So this is how simply plotting the Lineweaver-Burke plot at different concentrations of the inhibitor, we can get an idea of whether it is a competitive inhibitor or not. Okay. So a good example is if someone, nowadays this is quite common in the newspaper. In many states, alcohol is freely not available. So due to that, people drink all kinds of solvents that are usually containing methanol. So methanol is usually added to industrial alcohol to make sure people do not drink that, because methanol is poisonous while ethanol is not. So the commercial spirit, for example, if you go to a medical store and ask for surgical spirit for clean a wound or to clean up anything, to disinfect something, if you buy it, that comes not as pure ethanol. Instead, it will have methanol added a little bit so that it is poisonous and people don't drink. Okay. So let us say someone drank that kind of a spirit and they come to hospital. So how do you treat them? How do you get the poison out of them? So the way to do is, so why is methanol poisonous in the first place? So methanol is acted upon by alcohol dehydrogenase, an enzyme present in our cell, particularly in the liver, that converts methanol into formaldehyde. And formaldehyde actually cross-links proteins, and therefore our cells will not function. So it is a preservative. In your biology lab in schools, you would have seen biological specimens preserved in a clear liquid, and that clear liquid contains formalin. Formalin cross-links proteins and preserves the shape. But of course, when you cross-link everything covalently, no enzyme, nothing is going to function. So therefore, nothing is going to degrade and it is going to be preserved. So due to this production of formaldehyde, methanol is poisonous. Now, alcohol dehydrogenase normally in our system works on ethanol. Ethanol is usually converted into acetaldehyde by alcohol dehydrogenase. And methanol and ethanol compete as substrates for the same enzyme, that is alcohol dehydrogenase. So now, when someone has drunk methanol and the enzyme is attempting to convert that into formaldehyde, and if you want to inhibit it, you provide that person ethanol. So you put that person on a bed and perfuse a certain amount of alcohol into the bloodstream over a long period of time. Okay, so that the ethanol acts as a competitive inhibitor, and methanol's conversion to formaldehyde gets slowed down. Okay, so this is a good practical example of competitive inhibition. So here by providing ethanol, let us assume S is ethanol, you prevent inhibitor methanol binding to enzyme. And when you do this for a while, this eventually gets cleared by our detoxifying part of the metabolism. And once methanol is cleared below a certain level, like you are actually buying time, you are slowing down this formation. You basically detoxify and recover the person. So next inhibition we are going to see is uncompetitive inhibition. So uncompetitive means it is not competing with the substrate. Okay. Instead, it binds to enzyme somewhere else, but not to the free enzyme. Only when the enzyme is combined with substrate, the conformation change of the enzyme is such that now a site where this particular uncompetitive inhibitor binding is made possible. Only when the substrate binds, first enzyme, then substrate, then the inhibitor binds. So inhibitor binds to ES. So this is by doing this is going to affect the rate at which this combination is going to happen, as well as it is also going to affect the Km. Okay. So at a high substrate concentration, V0 approaches Vmax by this factor. So this factor exists in the previous one also, I kind of ignored it. So the Km increases by a factor of alpha, and alpha equals 1 plus concentration of the inhibitor divided by the this rate equilibrium constant for enzyme inhibitor formation. And then the Ki can be calculated as usual by the reactant by product concentration. So alpha Km is what the Km is modified by a factor of alpha in competitive inhibition. Whereas here you introduce two terms, alpha, only one in this next one, we are going to have two. So alpha prime, okay. And that is going to affect the Vmax. So this is primarily because as inhibitor increases, it is going to drive the forward reaction here to combine with the ES, because you are removing ES. So this forward reaction like K1 rate constant, this one will be enhanced. And due to that, the Vmax is going to decrease by that factor, and which means Km also is going to decrease because the substrate required to reach half Vmax is also going to decrease. So due to that, Km, Vmax, they both change by the same factor, that alpha prime. So the slope remains the same. Slope doesn't change. At increasing concentration of the inhibitor, slope does not change, but then the Vmax decreases. Okay, so yesterday I told you we are plotting reciprocal. So this direction means it's actually decreasing, and this direction actually means decreasing. So this shape tells you that it is an uncompetitive inhibitor. Then you have a situation where both are changed. They are called mixed inhibitors, meaning they can bind with the free or the substrate bound enzyme. It can bind with E as well as ES. And therefore, both of this come into play. So you have the competitive situation, that is alpha Km, as well as you have the uncompetitive situation, that is the alpha prime. So the actually full depth of this kinetics is quite complex, but we are not getting into all those details because it's an introductory course. So we are just familiarizing that there are certain factors by which these components of this equation, these terms are going to change, these parameters are going to change, and that will affect the shape of the reciprocal plot, and the characteristic shapes indicate the type of inhibition. So that's the main point we are getting here. So here you don't get parallel line. but at the same time, the slope changes. That indicates it's a mixed inhibitor. Okay, so this is all we need to understand. So if you want to get into good details, you can try to dig further in the references given in Leninger in this chapter to get an understanding of how these become the like how these terms come to be about divided by alpha prime, or alpha km divided by alpha prime. All those things you can try to work out the equations and the conditions by going through the back references if you really get very excited about learning about enzyme inhibition in detail. But for this particular course, what is here in the slide is sufficient. So you need to mentally, visualize these situations multiple times to get an idea of why the Vmax changes, why Km changes, all those things. So now we are going to look at, so far what we were looking at were all reversible inhibitions, where the binding is reversible, going by these equations. You see here, if the forward as well as reverse, both are possible. So these are all reversible reactions. So these are reversible inhibitions. And there are irreversible inhibitors. They covalently modify residues on the enzyme, oftentimes the active site residue, the residue that actively participates in catalysis. And they are useful to identify what residues are in the active site. For example, if you take this chymotrypsin serine hydroxyl group, serine 195, on the enzyme. 195 indicates the amino acid position. From the N-terminus, this is the 195th amino acid. N-terminus meaning the end of the molecule where that amino acid is, the amino acid's amino group is not in any peptide bond with another amino acid. Okay, while its carboxyl group is in peptide bond linkage with the amino acid number 2. And 3, 4, and so on. This is the 195th amino acid in chymotrypsin. And that is in the active site. So this binds to the active site. This is 2 isopropyl groups, or diisopropyl fluorophosphate. So when this molecule binds to this, this is highly reactive and it covalently binds to it. And once it binds, this is irreversible and this is not getting cleaved and the enzyme is permanently inactivated. Okay. So here you are not going to be able to reverse or change the kinetics by increasing the substrate. Okay. So these help us to get at the mechanism of the reaction. So for example, this helps in identifying that the serine is the residue at the active site participating in the reaction. And there are some versions of these inhibitors. They are called mechanism-based inactivators. So what they actually, and they are also called suicide inactivators. That is because they become reactive only when they bind to the active site. Remember all that we learned about transition state, induced fit, like the enzyme and substrate both undergo conformational changes such that the transition state of the substrate and the induced fit state of the enzyme are the perfect fit. And these suicide inactivators undergo that kind of distortion, and when they undergo to that distortion, then they become reactive and then they kill the active site. And that is why they are called suicide inactivators. And since it is mechanism dependent, the mechanism of the enzyme catalyzed reaction dependent inhibition, they are also called mechanism based inactivators. Okay. And understanding of these help in rational drug design. Okay. So drugs, I told you, are actually inhibitors of the enzymes. And that is an understanding of what kind of distortions they undergo and what readily binds to the active site irreversibly, etc. That knowledge helps in designing the drugs. So this is one of the reasons why people crystallize and solve the structure of the enzyme so that they know all the active site residues and the surrounding amino acids, and then you go ahead designing a drug that will fit there properly like a substrate, but then will inactivate the enzyme activity. Okay, so now we move on to another topic. With this, we will be done with enzymology, and this topic is how the enzymes are regulated. And some of the enzymes are constitutive. So far, whatever we saw, they are all constitutive. If they have substrate, they are going to act and produce the product. So their activity itself is not modulated or altered. Okay, and that happens, and when for some enzymes, and such enzymes are called regulatory enzymes. Most of the enzymes in our metabolic pathways were conversion of one starting molecule through a series of individual steps to a final product. For example, producing energy out of glucose involves a series of modifications to glucose. Glucose becomes glucose 6-phosphate, fructose 6-phosphate, then fructose 1,6-bisphosphate, and so on, finally to pyruvate. And the pyruvate enters the TCA cycle to produce energy. And that sort of long pathway, each step is catalyzed by an enzyme, and these enzymes are regulated based on whether the substrate is in abundance and product is in short supply, or vice versa. If you have too much of product, there is no point in converting the substrate again. Or if a downstream step is going slow, no point in going faster with an upstream step. It's like an assembly line. You want to regulate based on the supply demand, and therefore regulation becomes very important in biochemical reactions, in metabolism. And how the enzymes are regulated is what we are going to consider here. So they are regulated by 4 different ways listed here. A set of enzymes called allosteric enzymes have a characteristic regulation. We will consider them in some detail. And some of them are covalently modified to be active or inactive. And some interact with other proteins that regulate them. And some undergo proteolytic cleavage before becoming active. So first let us look at the allosteric enzymes. They are really important. A good example of allosteric enzyme is actually a non-enzyme, a hemoglobin. Many of you already know hemoglobin, and hemoglobin is the molecule. It's a protein. It's a tetramer. So you have 4 subunits, 4 polypeptides interacting together to make the full complete protein, and that has a prosthetic group, which is the heme, and it also contains a metal cofactor like ion present there, and this binds oxygen and transport oxygen from lungs to tissues in via our RBCs in the blood. Okay, so in hemoglobin you have 4 heme, and therefore 4 oxygen molecules can bind, and binding of oxygen to one subunit of these 4 subunit protein induces changes in the conformation such that the remaining substrates, subunits more readily bind oxygen, bind at a higher rate, and that is allosteric effect. Okay, so the allosteric regulator, okay, in this case the substrate itself, which is oxygen, so binding to one unit activate the substrate binding, that is oxygen binding, on the other subunits. So with respect to, let us say you are going to consider the subunit to which oxygen first binds as subunit 1. Now for subunit 2, the subunit 2's activity is enhanced by the binding of oxygen to subunit 1. So meaning the activator binds somewhere else other than the active site. Okay, here we are considering the subunit 2's active site. With respect to that, oxygen binding in subunit 1 is somewhere else in the protein. And somewhere else is what is this word allo. Steric is the position, other than the active site. So the modulator, which may be activator, in the case of oxygen and hemoglobin, oxygen is the activator because oxygen binding to one subunit activates oxygen binding in other subunits. And that sort of factors called the allosteric activators. And to consider it could be activator or inhibitor, we call allosteric modulator, okay, allosteric regulator. And that binds elsewhere. And these proteins like hemoglobin, in hemoglobin multi-subunits are there, but for one subunit, the other subunit's active site is like the regulatory subunit and the regulatory molecule allosteric binding site. But otherwise, it is an active site. So if you leave out hemoglobin and take a normal allosteric enzyme, they usually have, like hemoglobin, multiple subunits, but each subunit may not be converting a substrate into product. And that kind of a thing, hemoglobin kind of thing, we call it as homotropic allosteric enzyme because one subunit's active site acts as the regulatory subunit and the regulatory site for another subunit's active site based on active site driven reaction. So in a heterotropic allosteric enzyme, one subunit acts as a regulatory subunit. Okay, it doesn't do any catalysis. And there the regulatory molecule, the allosteric regulatory molecule, here indicated by M, binds. And that binding induces the conformational change in both the subunits, such that in the catalytic subunit where you have the active site, the shape changes such that now substrate readily binds. This kind of a situation very commonly we see when we learn about cell cycle regulation. So probably in a later course when you learn cell biology, you will learn about how cell division is regulated. There you will find a set of proteins called kinases are important for regulating the various events of cell division. And these kinases bind to another set of molecules called cyclins, and these cyclins act as the regulatory subunits. So cell division dependent kinases, CDKs, they are like the active site, like this C, and the cyclins are the regulatory subunit, like this R. And these cyclins regulate the activity there. So that is a real life example for this cartoon. But the primary point here is allosteric enzymes have multiple subunits, and if we consider two kinds among those subunits, one set of subunits are the regulatory subunits where the regulator binds, that induces conformation change in the active site such that now the catalytic subunit's activity is increased. Okay. On the other hand, a negative regulator will do the opposite. When it binds, the conformation changes such that the substrate doesn't readily bind. Okay, so both are possible. Positive regulation, negative regulation. In a situation like hemoglobin, where substrate binding to one subunit, activating substrate binding to another subunit, I told you these kind of enzymes are called homotropic enzymes. So these homotropic situations, when the effect is positive, we call that as positive cooperativity, because there is a cooperation among the subunits. When the substrate binds, oxygen binds to one subunit, it increases oxygen binding in the other subunits, meaning the subunits are cooperating in a positive manner because the binding is enhanced, because here the function is actually binding oxygen and transporting. So we call that as positive cooperativity. If the opposite happens, binding in one reduces the binding in other subunits, then you call that as negative cooperativity. Okay. And for these enzymes, okay, so this is before I get to the kinetics, we need to consider these. So these enzymes are large multi-subunit complexes. So an enzyme molecule, if you take each color here is one subunit. Like if I'm going to take this strong pink color subunit in the right image. So an individual subunit itself is a lot of amino acids. It is a very big molecule in when you consider molecules in normally encountered in organic chemistry. So these molecules are large. An individual enzyme, a polypeptide is large, primarily to ensure all those various interactions that we learned about when we learned binding energy, the idea of binding energy and how binding energy contributes to reducing activation energy. And for that sort of non-covalent interactions and orienting the substrate and desolvation and so on, to bring about all of that in a very stable manner, you need large molecules. That's why enzymes tend to be large molecules. These allosteric enzymes are an order of magnitude larger because they have multiple subunits, because some act as regulatory subunits, some act as catalytic subunits. And allosteric enzymes are often subject to feedback inhibition. So this is an important concept. So remember, metabolic pathways, we are right now studying the property of individual enzymes, but in our body, enzymes function in a series of reactions. Okay, I keep telling this, glucose getting converted to pyruvate through a series of steps. So this is what you are seeing here. This is an amino acid biosynthesis, a threonine becoming isoleucine through these intermediates, and each intermediate is catalyzed by one enzyme. So in the beginning of the enzymes, we learned about intermediate steps. Those are different. Those are actually from this to A or A to B in one reaction. The E2 converting A to B might involve multiple intermediate steps, and that is the intermediate steps we were learning initially. Now we are looking in a metabolic pathway, one set of molecules going through a series of intermediate stable molecules that are present in the cytoplasm stably and which could enter into other pathways, meaning these can be cross-linked. Those sort of intermediates, here A diffuses out from E1. So that E1 could have had multiple intermediate steps within its active site converting this to A, and that is the kind of intermediate steps we learned earlier. Now we are looking at a biochemical pathway where one product is the one substrate is converted into a subproduct which is released out of that enzyme. Okay, which can go into multiple different pathways, and in this particular direction, then E2 acts on A to make B, and so on. So in this sort of a multi-step biochemical pathway, oftentimes the very early step is catalyzed by an allosteric enzyme, and its activity is often subject to regulation by the ultimate product, not the immediate product of that enzyme. So the purpose of this entire pathway is to produce this. If isoleucine is present in large excess, like you ate food, when digested, it produced a lot of isoleucine. So the cell need not make isoleucine using its reserve threonine. So when you have a lot of isoleucine, then this entire step can be inhibited. So usually the first step, it makes sense. Why make A if you are not going to use A to make B to C to D to isoleucine? So you stop here itself, close the tap at the very first step. And that sort of end of the pathway product acting as a negative allosteric effector of the enzyme catalyzing the first step is a common thing. And this we call as feedback inhibition, because this is providing a feedback to this pathway. Okay. And allosteric enzymes often are subject to feedback inhibition. Usually the first step involves an allosteric enzyme, and that's usually negatively regulated by the end product of the entire pathway. So with this, I'll stop today's class, and we'll continue to, for the first set of molecules now we are going to see in detail are going to be carbohydrates. So that's what we are going to move to. So we had a brief introduction to the molecules. Then we had some good look on the enzymes, the biocatalysts. Now we really move on to understanding the reactions in the cell. And before getting into those reactions, let us familiarize in some detail about the molecules, and as well as principles that govern energy transformation in the body, and then we will go to the actual reactions. So that's the order we are going to go. So next we will move to carbohydrates in some detail in next class. So if you have questions, please ask. Otherwise, I'll see you next Monday.
Sir?
Yeah, go ahead.
So, what kind of substances are positive modulators made of?
What kind of?
Substances, like, are they like sedatives or something like that, the positive modulators?
Oh, okay. Positive modulator in a pathway. So you're wondering why would, for example, in this case, threonine stimulate its own conversion to isoleucine, right? So this negative feedback you readily understood. That is, if I have a lot of isoleucine, then I don't need to do this. So negative feedback I understand. So your question is, what kind of situation will need positive activation? So sometimes what happens is the substrate is in great abundance and that needs to be quickly metabolized. And in that situation, that high concentration of the substrate, if that itself can induce its conversion, and that helps. So that is positive feedback inhibition. So right at this moment, I only think of the oxygen and hemoglobin, but an actual metabolic enzyme, we will learn when it comes on our way. There are many that will come. So that is a situation usually the substrate itself activates the enzyme's activity.
Okay, yes, I got it. Thank you.
Okay. Any other question? Let me look at this. There is one question on the chat. Yes, yeah, so I got your question. Will the middle enzymes be affected? Yes, it depends on what else can happen to B. If there is a branching, if B can be used in some other pathway, then it is actually okay to convert threonine to A and move on. And this, to address this question, I need to evoke another concept called committed step. So rate limiting step you understood. Okay, the rate of that particular step limits the overall rate. There is something called committed step. Let us say formation of A from threonine is only for the purpose of making isoleucine, and A has no other fate, that's the word we commonly use, in the cell. That is, A is not useful in any other manner in the cell. In such a situation, making A is useless when you have a lot of isoleucine. And that kind of a step is called a committed step, meaning you are, by doing this reaction, you are committing this threonine to making isoleucine. And usually that is the step that is regulated allosterically. And in this particular case, usually committed steps are rate limiting also. But let us say in a pathway, A can actually participate in many things. Then A may not, E1 may not be subject to regulation. Instead, E2 or E3 may be regulated. So therefore, the feedback regulation may be on the other enzymes like E3 or E4 or any of them. Did this address your question?
Yeah, yes, sir. Thank you.
Okay. All right. See you guys Monday.